Good morning and welcome to the 2021 FDA Science Forum. I'm Mike Kaczynski and I will be your host today. We have two jam-packed days full of presentations. With that being said, we're going to jump into get started, and I'd like to introduce you to our first speaker, Sharon Watson. Sharon, go ahead and turn your camera on.
So good morning, everyone. My name is Sharon Watson. I'd like to welcome you to the 2021 FDA Science Forum. Before we get started today, I just have a few housekeeping and continuing education remarks. Continuing education is granted for this event. Please feel free to visit fda.gov for more information regarding CE for your preferred sessions for these 2 days. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than that of a single supporting company. Also, for our disclosed unapproved use, our speakers today are required to disclose to the attendees when products or procedures being discussed are off-label and any limitations on that information that is presented. The FDA CE consultation and accreditation team have nothing to disclose. However, the following faculty and planning committee members have disclosed information which you may view on the fda.gov website under continuing education. Requirements for receiving CE: physicians, pharmacists, nurses, and those claiming non-CME must attest to your attendance and complete the final activity evaluation via the CE portal. CE information will be emailed to participants. For multi-day activities, participants must attest to each day and complete the final activity evaluation for both days. For pharmacists and pharmacy techs, failure to provide your correct National Association of Board of Pharmacy information in the correct format may result in loss of credit for this activity. Your NABP profile number should be a 6 to 7 digit profile assigned by the CPE monitor, and your birth date should be in the month-day format. Do not provide your pharmacy license number. Just click on the My Account tab and then navigate to the Edit Contact Information to verify your information is correct. All attendees will have 14 days from the last day of this activity to log in, complete the required evaluations, and attest to your attendance to claim credit. You may then view and print your statements of credit. Pharmacists should log into the CPE monitor 8 weeks after the last session of your activity to obtain your CE credit. The objectives for our forum this year: one is to discuss contributions to the evolving science of clinical, non-clinical, and post-market evaluation, explain how artificial intelligence and big data together can improve public health, discuss how the FDA leverages social and behavioral sciences to empower patients and consumers, describe ways in which we're moving into the future, discuss the potential utility and challenges of new technologies such as microphysiological systems, microbiome, or a combination of both in advancing product development and integrating this knowledge in scientific communications with regulatory and research work. Discuss the application of innovative tools and approaches to support pandemic response, development and evaluation of medical countermeasures, and the detection of advantageous agents. Distinguish the varied levels of activity within the FDA and the various external stakeholders in protecting public health, and finally describe several types of regulatory science related to substance abuse, misuse, addiction, and how it can be used to inform FDA regulatory decisions. So without further ado, I would like to introduce the FDA Chief Scientist, Rear Admiral Denise Hinton.
Thank you, Sharon, for that kind introduction, and good morning, everyone.
We all know that an event of this size and scope is nearly 2 years in the making, and I'm sure that everyone involved with the organization and planning of this year's event has dedicated many months to highlighting FDA's groundbreaking science, even while meeting the prodigious demands of FDA's COVID-19 response. So before I begin, I want to express my appreciation to all of you. A special thanks to the Planning Committee, the co-chairs Roxari Shahizada and Sharon Watson, as well as to all of the Planning Committee members across the agency and the Chief Scientist Office of the Scientific Professionals Development, which helmed this event and does so much to support the training and education of our scientists. This year's theme, Science as the Foundation for Protecting and Promoting Public Health, is an essential reminder that science is at the core of everything FDA does. Over the next 2 days, you'll be hearing from national experts about the impact of globalization and novel technologies on the products we regulate. You'll hear from our own staff about the scientific research they are doing to understand and apply new science and technologies to our regulatory decision-making and other regulatory activities. But before I turn the podium over to our acting commissioner, I want to give you a sense of the ways in which the FDA's Office of the Chief Scientist supports and facilitates some of the cutting-edge regulatory science activities underway at FDA. This has been an unparalleled time to be part of the FDA. The COVID-19 pandemic has revealed, as has almost no other time in a century, the transformative effect on our lives of emerging technologies, new areas of science, and globalization. The tectonic shift created by the convergence of these elements in the field of regulatory science has produced both unrivaled challenges for the global scientific community and exciting opportunities for improving public health. Over a decade ago, Congress established the Office of the Chief Scientist to foster the robust scientific coordination and collaboration that FDA needed to respond to the transformations taking place in the 21st century regulatory landscape. And as the COVID-19 pandemic has proved, in an increasingly interconnected world, delivering on the promise of new technologies demands a collaborative response across FDA and with all stakeholders, domestic and international, in the regulatory science enterprise. I'd like to highlight just a few of the ways in which OCS has stimulated collaboration within the agency and with our stakeholders, most especially during the pandemic. And this is to modernize the scientific and technical tools to advance FDA's mission. The National Center for Toxicological Research in the Office of the Chief Scientist holds a unique and foundational position at FDA. It is the only center that supports all FDA offices and product centers and provides the essential toxicological research required to conduct those scientific activities. NCTR has made remarkable contributions both within the agency and with our domestic and international stakeholders during FDA's pandemic response. One NCTR COVID-19 investigation exemplifies the type of approach we must adopt if we want to prevent, detect, and respond effectively to future pandemics, natural disasters, and pandemics, as they will inevitably occur. Using a complementary tool for estimating the viral spread of COVID-19 in the central Arkansas area, NCTR's Jefferson Labs are surveilling SARS-CoV-2 in wastewater. Project goals are to establish a method to extract and quantify SARS-CoV-2 in wastewater samples. And they are to monitor the temporal dynamics of SARS-CoV-2 titer in wastewater as a proxy for the presence and prevalence of COVID-19 in Arkansas communities, and estimate the number of clinical cases based on the titers of SARS-CoV-2 in the wastewater samples. Early detection and continuous monitoring of COVID in the community will help federal and local agencies respond more quickly to identify and halt the spread of COVID-19 and decrease the burden of patient admissions in healthcare facilities. This is a tool with a direct public health impact. What's more, implementing routine surveillance should provide valuable information about the efficacy of interventions underway. As the COVID-19 pandemic emerged, my office also tapped into a network of academic centers of excellence in regulatory science and innovation. This is what we call the CIRCES. And this is what OCS established in 2011 to help our product centers leverage research expertise and resources not available in-house. Our Johns Hopkins University CIRSE recently investigated a highly underused statistical analysis methodology that we hope will expedite evaluation of potential drugs and biologics for treating and preventing COVID-19 by decreasing the number of participants needed in the clinical trial. One result of this project is a report on improving precision and power in randomized trials for COVID-19 treatment. The research simulated clinical trials based on data from hospitalized COVID-19 positive patients and concluded that covariate adjustment could speed up many types of COVID-19 treatment trials. In another example, the UCSF Stanford CIRSE has begun research on a rapid query model that is enabling us to examine prioritized COVID-19 questions using electronic health record data. The model enables COVID-19 questions to be quickly addressed using a UC Health Data Warehouse, which contains data from over 100,000 patients tested for COVID-19, with over 3,000 of them testing positive. Questions of recent interest include, what are the daily use patterns of remdesivir and various corticosteroids among the patients hospitalized due to SARS-CoV-2 from the UC Health Data Warehouse? We are using real-world patterns of medication use, vaccines administered, and comparative effectiveness analysis of various treatments for SARS-CoV-2 infection. Recognizing FDA's need for a nimbler contract mechanism for soliciting external expertise on specific research challenges, OCS also established and manages the Advancing Regulatory Science Broad Agency Announcement, or BAA. Well-established collaborations with our stakeholders supported by BAA contracts at home and abroad have enabled FDA to work swiftly in support of the application of new technologies to the development of therapeutics and vaccines for COVID-19. For instance, the BAA has been used to support multiple research projects funded by the Chief Scientist's Office of the Counterterrorism and Emerging Threats, which we call OSET, which provides critical support for FDA's national and global health security, counterterrorism, and emerging threats portfolios. The benefits of these existing partnerships have never been more apparent than during this pandemic. OCS's OSET is using BAA-funded contracts to support FDA medical countermeasure work on Ebola and Zika research, and they have been able to quickly extend these BAA contracts and apply their research methods to study COVID-19. Collaborations with Public Health England and Stanford University were expanded early in the pandemic to leverage the tools developed to assess the efficacy of Ebola vaccines and therapeutics to gather vital information about COVID-19 infection. And recently, in partnership with the National Institute of Allergy and Infectious Disease, NIAID, OSET has awarded a new BAA contract to the University of Liverpool, which together with a global consortium will sequence and analyze samples from humans and animals to create profiles of various coronaviruses, including SARS-CoV-2. The investigation will also use in vitro coronavirus models like organs on a chip or microphysiological systems to inform development and evaluation of medical countermeasures for COVID-19. With the goal of advancing new tools as well as new areas of science, OCS has formed cross-agency working groups in areas of high priority to the agency. FDA's Alternative Methods Working Group is a good example. The group serves as a catalyst to the development and potential application of alternative systems, in vitro, in vivo, in silico, and system toxicology modeling, like microphysiological systems, including organs on a chip, to inform FDA decision-making and regulatory toxicology. These tools will help achieve a goal FDA has long been working toward, to reduce and refine, even replace animal testing. And may also help bring novel FDA-regulated products to market faster while preventing products with increased toxicological risk from even entering the development pipeline. The working group has also launched a webinar series that gives developers a chance to present their cutting-edge methods and methodologies exclusively to FDA scientists. And so far, the group has held over 4 such webinars. Early this year, the working group published the agency's report, Advancing New Alternative Methodologies at FDA. And this highlights the significant progress FDA scientists have made in laying the groundwork for integrating alternative approaches into FDA regulatory programs. Many of the important research projects on alternative methods this report describes are being carried out in FDA laboratories, and many of these include collaborations with outside stakeholders. Results from these cutting-edge research activities add to the body of knowledge that FDA can draw on as it evaluates regulatory acceptance criteria for new alternative methods. I encourage you to read this report, and it can be found on the Advancing Alternative Methods at FDA section of our website. Time won't allow me to share with you all our achievements, but in closing, I just want to emphasize how vital the research you will be hearing about over the next 2 days is guiding FDA's work. In the coming decades, the speed of technological advances will only accelerate. To stay current, to be innovative, to continue to meet today's public health challenges and those of the future, FDA must and will continue to collaborate. We need to engage all our stakeholders to ensure that we fulfill the promises of new science, emerging technologies, and an increasingly interconnected world. And that is what the FDA Science Forum is all about. Thank you for your commitment of time today to learning about all the great work that we are doing. And now it is my pleasure to introduce the Acting Commissioner of Food and Drugs, Dr. Janet Woodcock. Over to you, Dr. Woodcock. Thank you. You have to come off of mute. Thank you.
Thank you. Yeah. Thank you, Denise, for the introduction and for everything that you do to lead our Office of the Chief Scientist and support the rigorous science at FDA. You know, the FDA scientific team is a remarkable group of individuals with such wide-ranging cross-disciplinary expertise that includes chemistry, biology, microbiology, toxicology, as you just heard, nutrition, veterinary science, dentistry, engineering, physics, pharmacy, and nursing. That's just a small sample, and we need all these disciplines because the regulatory role that we play requires science from almost every quarter of science, including, say, mathematics, to be integrated into our work. But this list demonstrates how well-equipped FDA really is to respond to the varied challenges, public health challenges and roles that we have regulatory responsibilities for. Today's Science Forum offers us an opportunity to highlight and pay homage to the work of these scientists. It also allows us to do a little well-deserved boasting about how well we're doing and to showcase some of the groundbreaking scientists themselves and their work that we call upon to inform regulatory decisions. Now, to give one very recent example, I'm delighted to note that our own Dr. Dion Price, who's Director of the Division of Biometrics 4 in the Office of Biostatistics and CDER's Office of Translational Sciences, was just elected the 118th president of the American Statistical Association to a one-year term that will begin in 2022. So congratulations to Dion. But it's just wonderful that our scientists are able to participate and are recognized by the outside world. Dr. Price herself has been involved in the FDA response to many public health challenges. She served on the Antibacterial Drug Development Task Force and a member of the teams reviewing products to treat Ebola and coronavirus infections. And she's currently leading a cross-cutting collaborative effort across FDA to better use complex, innovative trial designs in pharmaceutical drug development, which is a very thorny scientific issue. She also, I think, understands, having faced the challenges of being presented with data and interpretations of those data pertaining to the pandemic, just how critical it is that statistics be properly used, particularly during public health emergencies. I first got to know Dion when she shadowed me for a brief time here at FDA. And congratulations on this honor. But as I said, many of our scientists are recognized by professional societies and by the scientific community at large. And the FDA Science Forum affords the public a chance to get to know and better understand the work of scientists such as Dr. Price and some of the work being done in support of our mission. Nearly every aspect of the work we do at the FDA to advance consumer and patient safety and promote health relies on the need for rigorous data and good science. And yet, as prepared and informed as we are, one of the most challenging and also exciting aspects of science is that it's rarely static. And in fact, it's frequently on the cutting edge, which we work on, the cutting edge of application to humans. There's always a great deal of ambiguity and uncertainty. And our job is to kind of nail down that certainty because we have to make decisions. Our scientists are always looking forward, working to stay ahead of the curve by trying to apply the newest technologies and tools, incorporating the best possible data. And we've certainly seen that ethos on display over the past 15 months in the agency's extraordinary response to the COVID-19 episode. During that time, we've supported the development of authorization, emergency use authorization of 3 vaccines that have met our robust standards for emergency use authorization. The development of more than 610 COVID-19 drug development programs in the planning stages and more than 450 trials of such agents. There are 9 COVID-19 therapies currently authorized for emergency use, and there's 1 approved treatment, as you know, and there are more than 400 EUAs covering nearly 800 medical products for devices, including molecular diagnostic, antigen and serology tests, sample collection devices, PPE, and ventilators. So this was a massive amount of work that we've been doing. We've also used our scientific expertise to protect people from products that could be dangerous or sold with fraudulent claims to prevent or treat COVID-19. For example, veterinary products that were being improperly redirected for human treatment, which resulted in some serious illnesses and a fatality. And we developed a data analysis tool to track the incidence of the disease and its impact on the food system and help ensure the safety and reliability of all the supply chains for all the products we regulate. And even if we've responded to this public health emergency, we've continued to apply our diligent, science-based approach to the agency's ordinary but critical responsibilities to protect and promote health. To give you just one important example, we recently committed, with the full support of the administration, to advancing 2 regulations that will dramatically change the landscape of combusted tobacco products. One proposed product standard that would ban menthol in cigarettes, and another that would ban all flavors, including menthol in cigars, including little cigars, which we plan to publish as proposed rules within the next year. What all this work underscores and reaffirms is just how essential and integral science is to the FDA. Quite simply, good science is in our DNA. As I mentioned earlier, like science itself, the FDA cannot and does not remain static. To continue to be an effective health and consumer protection agency for the 21st century, we have to continually expand our scientific and data capabilities. We're only as good as the data we have to analyze and the science that we apply to it. We recently began an important effort to become better informed, more efficient, and better prepared by launching the Technology Modernization Action Plan, TMAP, and Data Modernization Action Plan, DMAP. These initiatives are designed to help us develop enterprise-wide technology platforms in support of our regulatory mission to make maximum use of data from current and new sources. I can tell you that this effort if we are successful in bringing enterprise-wide modern technology platforms and then consolidating and enabling use of the data that we now have stored in siloed systems and unfortunately in some cases in file cabinets. This will really get us to a higher level. In addition, we're going to continue to focus on hiring and retaining the best scientists to ensure we can continue to operate at world-class levels. I think the good news is this effort will find increasing public support as more Americans today have a better understanding and appreciation of the importance of science in their lives. And I can certainly say that for the legislature. One recent survey found that global confidence in science has hit a new high. The poll was conducted by 3M during February and March of this year and interviewed thousands of people across 17 countries, including U.S., Brazil, Germany, India, China, and Japan. It found 91% of people trust science, up from 86% when the poll was first conducted 3 years ago. Of course, there are loud voices out there, very loud, that say they don't trust science, but the poll is reassuring that actually there is widespread trust in science in the human population. And certainly part of the increase right now is due to the public's reliance on scientists and health experts to explain and get us through the current pandemic. I hope that this awareness also includes an understanding of how critical it is that agencies like the FDA have the resources, capacity, and scientific independence needed to continue conducting the state-of-the-art scientific research, evaluations, and inspections that are necessary to keep the public healthy and safe. And credit for this increase, I think, in the appreciation of science goes to public health scientists here at FDA and our sister agencies at HHS and elsewhere who've done such incredible work. And I would suggest there's been another important catalyst, at least in the United States over the past year. One individual has been particularly influential in helping spur this greater understanding and appreciation for science. And I'm delighted and honored that he's here with us today as our guest speaker. As you all know, Dr. Anthony Fauci has a long and distinguished scientific career. I encourage you to take a look at his full bio or read one of the many hundreds of profiles that have been written on him over the past year or so. I'll give you just a few of the basics. He was appointed director of NID in 1984, and in that role he continues to oversee an extensive portfolio of basic and applied research to prevent, diagnose, and treat established infectious diseases such as HIV/AIDS, respiratory infections, diarrheal diseases, TB, malaria, and emerging diseases such as Ebola and Zika. Dr. Fauci has advised 7 presidents on HIV/AIDS and many other global and domestic health issues. He was one of the principal architects of the President's Emergency Plan for AIDS Relief, PEPFAR, a program, of course, that CEDAR and we at FDA participate in that has saved millions of lives throughout the developing world. And as a rheumatologist, I'm particularly aware of Dr. Fauci's contributions to immunology. As part of his work on immunomodulation, he developed effective therapies for formerly fatal inflammatory and immune-mediated diseases such as polyarteritis nodosa. His book on the vasculitides was often in my hands in the clinic, and that was a reference I frequently refer to. Dr. Fauci is the recipient of numerous prestigious awards, including the Presidential Medal of Freedom, which is the highest honor given to a civilian by the President of the United States, and the National Medal of Science. I could go on and on, but perhaps more significant today than these scientific achievements is the role of international discourse on issues of public health. That's because more than anyone, Anthony Fauci exemplifies the essential importance of letting science be the key determinant in answering and responding to the public health challenges we face. His extraordinary ability to help translate complex issues into common sense answers has helped the public understand that the scientific answer is also the most rational one. He's also the guy who the American public trusted to tell the truth about COVID and the most effective ways to deal with it. It's no small achievement for such a high-achieving scientist that nearly every description you read or hear about him includes phrases like straight-talking, down-to-earth, common sense, accessible, and of course, Brooklyn accent. I've had the opportunity to work with Dr. Fauci over the years, including most recently in their work on Operation Warp Speed, and I can say it's been a privilege and an honor. He's not just led a response to the pandemic, but at the same time, he's helped to make science appreciated, and dare I say it, even cool in some quarters. Not since the days when posters of Albert Einstein graced college dorms have we seen this enthusiasm for and humanizing of scientists and science, particularly those committed to protecting the public health. I'm sure you've all seen the pictures that dot lawns with the message, "Thank you, Dr. Fauci." But that's just the start. I recently saw that one company's making Band-Aids with his picture. They're called Fauci Ouchies. That's appropriate, given that Dr. Fauci helps protect America like a Band-Aid protects a person. For all these reasons, I can't think of a more appropriate speaker for the FDA Science Forum. And so it is my great pleasure to be able to welcome Dr. Anthony Fauci to the FDA and the FDA Science Forum. Thanks.
This is Tony Fauci here at the NIH, and I'm pleased to speak with you today virtually at the 2021 FDA Science Forum. As you can see from this first slide, I'm going to be discussing lessons that we have learned and the remaining challenges associated with the COVID-19 pandemic in 2021. I'm dividing the discussion into 7 components, and I'll briefly touch on the highlights of each of these. So first, starting off with the epidemiology. As we all know, we were confronted with a brand new virus at the end of 2019 when unusual cases of pneumonia were noted in the Wuhan district of central China. Soon thereafter, the Chinese identified and put on a public database a new strain of coronavirus as the source of this outbreak. Little did we know at that time that this would evolve into what historically is the worst pandemic that our planet has experienced in well over 100 years. With the numbers shown on this slide, with over 165 million cases and almost 3.5 million deaths globally. The United States, unfortunately, has suffered greatly, one of the top 3 countries in the world with regard to cases and deaths per population number. As shown on this slide thus far, there have been over 32 million cases and just short of 600,000 deaths thus far, and we are not at the end of this yet. Our country has experienced 3 major surges since the beginning, the slopes of which are demonstrated on this slide, the worst of which was in the late fall and early winter of the end of 2020, the beginning of 2021. We have since plateaued, and as shown on the right-hand part of the slide, we are now starting to see a gradual but definite diminution in the average number of daily cases. This is obviously related to vaccinations, which I'll get to in a moment, but this is going obviously in the right direction. Some interesting aspects of the epidemiology is the racial and ethnic disparities, particularly among African Americans, Latinos, and Native Americans, where their incidence of infection and the incidence when infected of a poor outcome related to underlying medical conditions. Moving on to virology. This is a beta coronavirus, an RNA virus with a large genome. There are 4 structural proteins, the most important and well studied of which is the spike protein with its receptor binding domain, allowing it to bind to ACE2 receptors on cells distributed in the upper, lower airway, the GI tract, and a variety of organ systems, including the cardiovascular system. We know a lot about the transmission now. It's transmitted by exposure to respiratory fluids through the upper airway by the inhalation of small, fine droplets, as well as aerosol particles. And these droplets or particles can be deposited on exposed mucous membranes in the mouth, nose, or eyes, much less commonly through contact with contaminated surfaces. The risk of transmission is greatest when people are close, but due to aerosolized, particularly with poor ventilation, there can be infection at greater distances than that. Other important issues with regard to this particular virus, that at least a third and as many as 40% of individuals never develop symptoms. A truly unique and unusual situation for a virus that can actually and has killed so many people. In addition, between 50 and 60% of all the transmissions occur from someone who either will never develop symptoms or a person during the pre-symptomatic stage of their own infection, complicating greatly the capability of doing identification, isolation, and contact tracing. The fundamentals of preventing acquisition and transmission now, first of all, and predominantly, is vaccination, but also public health measures such as wearing of masks, maintaining physical distancing, avoiding crowds and congregate settings, particularly indoors, and performing activities outdoors much more preferentially versus indoors, and the simple thing of frequently washing one's hands. Diagnostics for this infection are three fundamental categories. One of the molecular tests, which are highly sensitive, a PCR to detect the actual virus, usually in the upper airway, nasal or nasopharyngeal swabs. The other is antigen testing, used much more for surveillance, less sensitive, but is now being increasingly used for broader surveillance among different populations. And then finally, antibody tests to determine if in fact one has been exposed and infected with the virus. The clinical course is multifaceted. First, the clinical presentation. Looking at the slide, you can see that many of the signs and symptoms are very reminiscent of a flu-like syndrome. One of the unique aspects of this particular infection is that in a substantial proportion of people, there's the loss of smell and taste, which generally precedes the onset of the respiratory symptoms. As I mentioned, a substantial proportion of people never develop symptoms, but those who do, about 80% of them have mild to moderate symptoms, and about 20% or so have severe or critical, leading to a case fatality rate of approximately 2.3% overall, but for those individuals requiring mechanical ventilation, the mortality can be as high as 20%. For those who have manifestations of severe disease, it is generally dominated by the acute respiratory distress syndrome. However, as we now get more experience with this disease, we can see that it can be multi-system in its involvement, including cardiac dysfunction, neurological and kidney disorders, and a very interesting and peculiar hypercoagulable state characterized by microthrombi in small vessels, as well as a acute situation, which is seen not infrequently, of thromboembolic phenomenon. There's a multi-system inflammatory syndrome, first noted in children, but also can be seen in some adults. It's rare, but when it occurs, it can be very serious, very reminiscent of Kawasaki disease. The people who are at increased risk for severe COVID-19 illness are older adults. If one looks at hospitalizations and deaths, clearly those elderly individuals, 75, 80, 85 years old, have a much, much higher incidence of hospitalization and death compared to younger individuals. But also people of any age with certain underlying medical conditions, diabetes, hypertension, obesity, chronic lung disease, chronic liver, kidney disease, also are at a much higher risk for a poor outcome, resulting in hospitalization and death. There is now a syndrome which is being much better and more characterized called post-COVID-19 infection syndrome, long COVID or post-acute COVID sequela, a number of different names. What it really is that approximately 10 to up to 30% of people who recover virologically from SARS-CoV-2 and COVID-19, but who have the persistence for as long as 9 months or even longer of a constellation of signs and symptoms which are generally characterized by profound fatigue, muscle aches, temperature dysregulation, dysautonomia, sleep disorders, and what people have described as brain fog, or an inability to focus or concentrate for any long period of time. A number of therapeutic interventions have been developed. For example, the direct antiviral drug remdesivir in clinical trials has shown to diminish the time to recovery. This has been approved by the FDA. Other interventions on the basis of clinical trials have shown enough efficacy to be given an emergency use authorization, such as various combinations of monoclonal antibodies, a bit controversial as to the efficacy of convalescent plasma, still having reports coming out most recently. But then also, we must realize that the therapy for early versus late disease is really much different, with therapy concentrating on the virus in early disease and therapy concentrating on dampening the aberrant inflammatory or immunological response, which is the reason why the common drug of dexamethasone has become the standard of care, particularly for people requiring respiratory assistance and or high flow oxygen. The NIH has established a treatment guidelines panel of a large number of experts with considerable experience in taking care of people with COVID-19. They meet regularly and have produced a living document which is accessible online, shown here on the red bullet, COVID-19 treatment guidelines.nih.gov, and clinical data from published clinical trials that have been in preprint servers, as well as expert opinion. This has been widely used throughout the world to guide clinicians for the treatment of this disease. Looking forward in the future, a considerable effort is trying to fill an important gap with regard to COVID-19 treatment, and that is effective direct antiviral drugs to prevent people from progressing for the need of hospitalization. This is very similar to the effort that was done in a very, very successful manner with the development of combinations of drugs for HIV/AIDS, antiviral drugs that have really transformed the lives of persons living with HIV. The same holds true for targeted drug development for hepatitis C. So this is something that we're going to be putting a major effort in, to design drugs to inhibit vulnerable targets in the SARS-CoV-2 replication cycle. This is a slide that's similar to the slide that I've shown very often with regard to the replication cycle of HIV. In this case, we're looking at the replication cycle of SARS-CoV-2. And as you can see, a number of vulnerable targets for things like entry inhibitors, protease inhibitors, and polymerase inhibitors. And we'll be hearing more about that as the months go by. Finally, the major success story in the challenge that we faced from COVID-19, and that is in the arena of vaccines. The success of this has really been quite impressive, to the point where Science magazine has deemed the development of COVID-19 vaccines as the scientific breakthrough of the year of 2020. I had written an editorial in Science last month to try and explain and answer the question that people often ask: how could it be that when we talk about the development of vaccines, that it usually takes multiple years from the time one identifies a new infectious agent to the time you have a safe and effective vaccine? And with COVID-19, the virus was first realized, as I mentioned on a prior slide, in January of 2020, and 11 months later, in December, we had a highly, highly efficacious vaccine going into the arms of individuals 11 months later. That was not because we rushed things. And as I stated in the editorial in Science, the speed and efficiency with which these highly efficacious vaccines were developed, together with their potential for saving millions of lives, were due to an extraordinary multidisciplinary effort involving basic preclinical and clinical science that had been underway out of the spotlight, under the radar screen, for decades before the unfolding of the COVID-19 pandemic. And some of these are the basic preclinical and clinical research to develop new vaccine platform technologies, particularly the highly successful mRNA. Work done at the NIAID Vaccine Research Center under the direction of Barney Graham, namely the development of an optimal immunogen in the form of the prefusion conformation of the spike protein, which was stabilized by certain mutations to allow it to be used as a successful immunogen. And then the extensive domestic and international clinical trials network, which we developed at NIAID for both HIV clinical trials and influenza, were in some respects repurposed to be used as the clinical trial network to test the vaccines. As you know, the United States government has partnered with either development and or facilitation of the testing of 6 companies involving 3 separate platform technologies: the mRNA, the chimp and human adenovector, and recombinant proteins with adjuvants. 3 of these, the Moderna and the BioNTech Pfizer mRNAs, as well as the J&J, have already received emergency use authorization and have been distributed to tens and tens of millions of people in this country and more than hundreds of millions worldwide. Other candidates, including Novavax and AstraZeneca and Sanofi and others, are currently in the process of gaining data to be able to ultimately submit for an emergency use authorization. Now let's take a look at the results with these vaccines. First, they were highly efficacious in clinical trials. As I mentioned, as shown in these New England Journal of Medicine reports, the Pfizer BioNTech vaccine, which was tested in 44,000 people in a randomized placebo-controlled trial, has shown a 95% efficacy. The Moderna vaccine, tested in 30,000 people, similar efficacy of 94.1%. The J&J or Janssen vaccine, which was tested simultaneously in the United States and in several countries, including South Africa, with their problematic variant, has shown a 72% efficacy in the United States, but a very high degree of efficacy against severe disease leading to hospitalizations and deaths. One of the really interesting things is that the effectiveness in the real world setting was as good or even better than the efficacy in the clinical trials, something that is somewhat unusual when you're dealing with an intervention and comparing the pristine conditions of a clinical trial with the real world effectiveness. These are some of the reports documenting this. On the left, a report from the University of Texas Southwestern and over 23,000 employees showed a 0.05% infection rate among fully vaccinated employees, dramatically lower than unvaccinated employees. Also, a number of studies looking at both Moderna as well as BioNTech, showing again a considerable amount of effectiveness when compared to unvaccinated people. This was not only in the United States, but a very important publication from Israel this past month, in which the data using national surveillance showed that this vaccine at every stage of disease was highly effective, including the B.1.1.7 variant, which is dominant in the United States. And as you can see, whether it's all infection in general, asymptomatic, symptomatic, hospitalization, or deaths, the effectiveness was highly, highly good in this sense, more than 95% in most cases. This was true for all ages. As you can see on this slide, even the elderly, greater than 75 and 85 years old, had efficacy hovering around 94 to 95%. Shown in a rather simple diagram, what happened in Israel was that in the red, you had cases continuing to go up as we began to see people vaccinated, but you reached a critical point where the lines crossed, where I believe we are almost at that point right now in the United States, whereas the vaccines continued to go up. At this point, it was around 61% of the Israelis had at least one dose. The number of cases dramatically plummeted to the point of being at a very low level. In the real world effectiveness, again, for the J&J, the Ad26, very similar to the kind of situation we've seen with the mRNAs. Finally, the vaccines are clearly very safe, as shown by a number of parameters, both the clinical trials and the expanded safety monitoring systems, both of the CDC and the FDA. Importantly, the vaccines that we are now using in the United States have had a positive impact on viral variants, even though the effectiveness of the antibodies induced by the vaccines are diminished somewhat by some of these variants. For the most part, the protection is good. So if you look at the variants that we're dealing with, the one dominant in the United States is the B.1.1.7. Less so are those that are troublesome, namely the 351 in South Africa and the 617 in India. But when you look at studies, for example, from Qatar, which is using the BioNTech Pfizer vaccine in a country in which you have both the B.1.1.7, the variant in the United States, as well as the troublesome South African variant, not only was the effectiveness good against the dominant B.1.1.7, but the 351, it has 75% effectiveness, but importantly, virtually 100% effectiveness against severe, critical, or fatal disease, even though, as we know, there's been considerable concern about the 351. Other studies, when you look at the BNT, namely the BioNTech Pfizer vaccine, also the degree and the height of the neutralizing antibody response showed that there was considerable effectiveness against a number of variants, including the 526 from New York, the 429 from California, and the 117, which is dominant throughout the country. Again, other studies, in this case showing the Moderna vaccine, again, the amount of antibody induced by the vaccine covered quite well variants B117351429. And the durability was considerable for more than 6 months. And again, if you look at the 617 variant, the one that's now dominant in certain regions in India, again, the modest neutralization resistance to this suggests that the current vaccines that we are using would have a considerable degree of protection, even against the 617. President Biden has made it clear that the goal in the United States is to get 70% of adults vaccinated with at least one dose by the 4th of July. And we hope by that time, the level of infection in the community will dramatically diminish, similar to the slide that I showed you regarding what happened in Israel. So at the end of the day, this is going to be and is a race between a highly, highly effective vaccine and the virus itself, which left to its own devices clearly would want and would surge. I believe we're at that critical point where we are going to see this race, if we continue to vaccinate people at the rate we're doing, that the vaccine will in fact win this race against this outbreak, and that within a reasonable period of time, hopefully, as we get into the summer and early fall, we'll begin to approach a degree of normality that we all have been longing for a long time. Thank you.